Welcome to Chapter 5, Microbial Mechanisms of Pathogenicity. This chapter fulfills objective number 4, specifically interactions between microbes and their host, microbes and human disease. We're going to be covering host entry, or how the organism gets into the host, penetration of host defenses, damage to host cells, pathogenetic properties of viruses, pathogenetic properties of fungi, protozoa, helminths, and algae, and portals of exit. First off, host entry. We're going to talk about portals of entry, numbers of invading microbes, and adherence. Portals of entry are basically any openings that lead into the rest of the body. So as you can see, these are all things that you're familiar with. The placenta, if you're a fetus, that's pretty much the only way that a pathogen is going to get to you before you're born. Now you'll notice that most of these are mucous membranes, okay? The moister portion of the skin. Most pathogens cannot get past intact skin. So skin is the other route. It's not as good of entry portal as the mucous membrane is, because the skin is dry and is thicker than the mucous membrane. Now, peritoneal route is any time you have an intentional break in the skin. So, for example, if you are undergoing surgery or you're getting an injection, that's considered a peritoneal route, which in effect is pretty much the same as broken skin or an insect bite. It's just artificial as opposed to natural. Now, all microbes have a preferred portal of entry. Now, you're already familiar with this. If a pathogen is going to cause intestinal distress, does it want to come through your respiratory system? No, it wants to come through the mouth. Okay. And for respiratory diseases, does it want to go through broken skin, or does it want to come through the nose or the mouth or the eye? Makes sense. Portal of entry, preferred portal of entry makes sense with the organism and what type of disease it's causing. Now, preferred portal of entry leads us to infectious dose, which is the numbers of invading microbes that you need for 50% of a sample population to become ill. So when we do these studies, our sample population is usually mice. So what we do is we take 50 mice, generally it's 100 mice, and we infect them with the pathogen, and we put in known numbers of infectious organisms until that brings us up to 50 of the mice are going to get sick. Now, we've also done studies in humans for the non-lethal organisms that only infect humans. Then for those that are fatal, we've done retroactive studies where we're not actually infecting people. Now, for an actual example. Shortly after the horrible events of September 11th, we had anthrax, weaponized anthrax spores going through the mail system. And about 50% of the people who were exposed to this poof of lovely spores when they opened up their letters, about half of them got respiratory anthrax, about half got cutaneous. So the question is, if you have this cloud of millions of spores, probably billions, because to be able to see, by the way, when I say spores, I mean what? Endospores. When you're in this cloud of probably billions of anthrax endospores, why didn't most of the people get respiratory anthrax? Well, here's the answer. The infectious dose for cutaneous anthrax is only 10 to 50 endospores. So if you had a fresh paper cut from opening up the letter, you didn't have to get as many endospores into the cut to cause cutaneous anthrax. With inhalation anthrax, or the type that leads to really bad pneumonia, you would need to get 10,000 to 20,000 endospores. That's why it was about 50-50. Then to get ingestion anthrax, which believe me, you really don't want to get, you need 250,000 to 1 million endospores. So what's the preferred portal of entry for anthrax? cutaneous. It wants to come through breaks in the skin. Now, for a pathogen to gain entry, it's got to be able to grab a hold. Think of it this way: if you're trying to get through a door with your arms full and there's a doorknob, are you going to get through? No, you got to free up a hand so you can grab doorknob, turn it, open the door. It's kind of the same thing for pathogens. They've got to be able to grab a hold of the host cell. So we call this adherence or adhesion. So to be able to do this, a pathogen needs to have molecules that we call variously adhesions or a ligand, and they match up with the receptor on the host cell, and generally they're very specific. Okay, so you've got to have both of these for the pathogen to be able to grab a hold. Now this explains the preferred portal of entry. So for example, we've got some E. coli that are grabbing a hold of human urinary bladder cells. They have ligands that match up with receptors on the bladder cells. Okay, if their ligands do not encounter receptors, say on human lung tissue, if human lung cells don't have receptors for E. coli, then you're going to need more E. coli to be able to cause disease coming through a respiratory route, whereas E. coli cause all sorts of bladder infections. That's one of their preferred portals of entry, is up the urethra. Here's a good spot to bring up the concept of avirulence. You have certain strains of a pathogenic species that through mutation can lose the gene for their ligand. Okay, and this is one cause of avirulence. You have a strain of a pathogenic bacteria that are not pathogenic. They are avirulent. They do not cause disease. Now, you can have this show up in more than just losing your adhesion. And we'll talk about some of these as we go through. You can also have a host that is more resistant to a particular pathogen by having a mutation where their receptor is changed so that the ligand or the adhesion doesn't grab a hold as well. Or they have a mutation where they don't have that receptor. The host receptors are not really intended for the purpose of being a doorknob for the pathogen. The pathogen uses proteins on the surface of the cell that are doing different jobs. So maybe that cell won't function as well without that receptor, but it functions a whole lot better than its buddies who are being killed by pathogens. So we can see how there's an interaction between the pathogen and the host that is mediated by mutations. Yeah, I had to bring in Chapter 8. This brings us to the next topic, penetration of host defenses. We're going to talk about capsules, cell wall components, enzymes, antigenic variation, and penetration in the host cell cytoskeleton. So, for penetration of host cell defenses, capsules, and cell wall components, most of this we've already talked about. Capsules, you should know how capsules aid in penetration of host cell defenses, which is, that's right, it makes the pathogen harder to phagocytize. Mycolic acid, let's skip to the bottom because we've talked about that. Mycolic acid performs approximately the same function as a capsule. It makes the cell slippery, so to speak. Okay, let's talk about the middle ones because we haven't talked about those very much. Fimbriae, remember, clear back to Chapter 3 when we talked about fimbriae. Fimbriae are rod-like extensions from the surface of a bacterium. Often they are ligands. They aid in the attachment of the pathogen to cells. If it's not a fimbriae, then it would be attachment proteins or adhesions. Well, the way that the pathogen gets past host defenses is to only produce these when they're needed. Okay, so say for example, E. coli only produce fimbriae for attachment when they're in the human gut. They don't produce them elsewhere. So that means that if you have antibodies to that fimbriae adhesion, then those antibodies are not going to be effective in neutralizing the pathogen, those specific antibodies, because the pathogen's not producing them. Okay, so they alter when they produce these various components. Okay, for this next section, enzymes, I'm going to present this as a little story so that you can see why a pathogen would make these different enzymes. Okay, so say I've cut myself and I have a pathogen that has entered into the deeper portions of my skin. Well, the surrounding cells are going to release warnings saying that, okay, there's been a breach of the defenses, i.e. skin, and we probably have pathogens. Well, these pathogens, if they produce coagulase, it will cause platelets to form a clot around them. Then when the white blood cells come, they say, hey, okay, we have a break in the wall, but I don't see any pathogens, so everything's okay. Then later, when the white blood cells leave, then the pathogens can produce streptokinase, which will break up the clot, they get into the bloodstream and wreak havoc. So you can see why it would be a benefit to the pathogen to produce both coagulase and streptokinase. That's a way of penetrating host defenses. They're hiding from white blood cells. Now here's another situation. Now suppose, this would be, in this situation, we've got epithelial cells, okay, lining the mucous membrane. And we always have a basement membrane. This keeps stuff out. Well, a pathogen grabs a hold of these epithelial cells and they produce hyaluronidase. That breaks down hyaluronic acid, which holds cells together. That's the type of acid that keeps this cell kind of glued to the next one. So if it can break down that glue, it can get through, break down the basement membrane, and get into the deeper tissues. So that would be penetration of host defenses by actually breaking through the barrier of, in this case, mucous membrane. Okay, antigenic variation. This is where a pathogen changes its proteins on the surface that your immune system would recognize. So on a consistent basis, it would just change those either through mutation or through reassortment or turning on different genes. Now, up here we have micrographs of flu virus, specifically H1N1. We have these little spiky guys sticking out of the lipid membrane, out of the envelope of the virus. You have the H antigen and you have the N antigen. They mean other stuff that you don't really need to worry about. But these vary. And because they vary enough that your immune system has to create completely new antibodies to combat the virus. That's why you get a flu vaccine shot every year, because of influenza antigenic variation. Now, we got all excited about the H1N1 flu virus because we hadn't seen those antigens since the great influenza of I believe it was 1917 through 1918, somewhere in that range. It was the great influenza that was the first recorded pandemic, a disease that swept through the entire world. So when we saw that antigenic profile again, we got really excited. And now in current flu vaccines, we have the vaccine cover H1N1 because it's currently, that is an antigenic variation that we're currently being exposed to. Now, penetration in the host cell cytoskeleton, this is stuff that we're looking at currently, which is kind of interesting. Some bacteria can actually attach to a host cell and tell it to phagocytize it on purpose. Or you can have changes in the cytoskeleton so that, such as in the case of hemorrhagic E. coli, you can form a pedestal. The host cell kind of forms a nice little extension that looks just like a pedestal. The E. coli sits on top of it like a king on a hill and causes death and destruction all around. This is a case of the pathogen talking to the host cell and basically confusing it into thinking that it's something else. So interesting stuff. That's about as far as we're going to go with that one for this class. Now let's move on to damage caused to the host cells. We're going to talk about cytophores, direct damage, toxins and superantigens, plasmids, lysogeny, and pathogenicity. So cytophores, okay, are proteins that carry iron. When you're thinking about that with humans, you usually think of hemoglobin. Hemoglobin does carry iron, but we're usually thinking about this in regards to how do you get the iron to the bone marrow where red blood cells are being produced, where you're making the hemoglobin. Well, cytophores are molecules that will carry these, and they're produced by the pathogen to grab a hold of the iron tighter than your carrier proteins. Okay, so it's kind of a tug of war. We want to use the iron for our purposes, the pathogen wants to use it for their purposes, and if the pathogen stills enough iron, then that causes damage to the host because we don't have the iron that we need for transport of oxygen and also for other enzymes. That leads us to the topic of direct damage. Not only are pathogens stealing iron, they can steal other nutrients. And they can also attach to a host or enter a host and take the nutrients from the inside so that the host cell can't use them. Makes sense. If a cell's being starved or if its structural proteins are being broken down, that causes direct damage. Also, with several pathogens that replicate inside of cells, when they've produced enough of themselves, rupture the cell and spread to the outside to infect other cells. That's definitely direct damage when you are excluded. Now let's talk about toxins. There are two basic types, the exotoxin and the endotoxin. Exo for out, are toxins that the pathogen creates and releases to the outside. Now, exotoxins, think of it as things that the pathogen are releasing purposely to damage cells and chew up cells so that the bacteria can eat the nutrients of the cells. Bacteria don't have teeth. They can't take bites of the host cell, so they have to release exotoxins. Endotoxins, on the other hand, are toxins that are a part of the cell that are released when the cell dies or when it is dividing. It usually loses some. Now, the most common exotoxin is the lipid portion of the lipopolysaccharides, LPS, that are part of the outer membrane of the cell of gram-negative bacteria. That's part of why I made you memorize what lipid A is, because it's an endotoxin. Damages the host. Now let's take a look at some specific examples of exotoxins. Okay, now in lab, you're familiar with at least one type of exotoxin, hemolysins. These are exotoxins that break down red blood cells. Let's talk about another one. Clostridium tetani produces the tetanus toxin. That is an exotoxin that is specific for human neurons. Well, they're specific for any neurons, but we'll stick to humans. And it prevents communications, normal communication between different neurons, and it causes muscles to contract and to not relax. That is an exotoxin. It is released by C. tetani. Endotoxins, okay, let's go back to our gram-negative bacteria. This is a situation where we're going to talk about the endotoxin, LPS, in the same category as superantigens, because LPS, lipid A, is a superantigen. When your immune system sees lipid A, it kind of goes nuts. It overreacts. We're going to talk about this more when we talk about inflammation in the next chapter, but way to think about it for right now is your immune system overreacts and causes such a strong immune response that you go into shock, you have a drop in blood pressure, you have respiratory failure, and you die. So for this reason, when you have a gram-negative infection, we have a tendency to treat it differently than we do gram-positive infections, such as your Clostridium. Because if you kill too many too fast, you release too many superantigens and it causes problems. This is also why gram-negative infections can cause problems faster than gram-positive infections, because if there's enough of them dividing, they're releasing the superantigen, and you get an over-response of your immune system anyway. Now let's talk about how pathogens get genes to make them more virulent, to make them stronger pathogens. Let's talk about plasmids first. You're familiar with plasmids. These are small circles of DNA that are outside of the bacterial chromosome. Actually, they show up in things other than bacteria, but we're going to stick to bacteria for this discussion. And you remember that they're passed from bacterium to bacterium through conjugation. That is horizontal gene transfer. And plasmids carry on them not only the genes for their own replication and spread via the conjugation pili, but sometimes they carry other genes, and some of those other genes can be the toxins or capsule proteins genes. And this can make the pathogen stronger. So think of it as being bitten by a genetically modified spider, and now you're Spider-Man. Da, 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 da. Except for instead of making superheroes, plasmids make super villains. So let's go on to lysogeny. Lysogeny, it deals with viruses. Some viruses, when they inject their DNA into a bacterium, rather than cutting up the bacterial chromosome and killing the bacterium, they insert their DNA into the bacterial chromosome. Then when the bacterial chromosome replicates, it makes more of the virus. The virus spreads slowly through a population, not causing any harm, but itself is being replicated. Now, it's kind of the same thing as a plasmid. They can carry genes for other than their own benefit. They carry genes for toxins and for other things that make the pathogen more virulent. Okay, so those are examples of how a pathogen can become stronger through infection. Doesn't seem fair, does it? Okay, let's talk specifics. Pathogenic properties of viruses. Viruses evade host defenses by antigenic variation, and also by not ever hitting the bloodstream and moving from cell to cell. That's one of the ways that viruses evade host defenses. Now, they also cause damage to the cells. Now, some of this is obvious. If they're taking over a cell and causing the ribosomes to make viral proteins instead of cellular structure proteins, the host cell is going to get sick. And some viruses spread by popping the cell. That definitely kills it, definitely damages it. Now, when we look at viruses in cell culture, we look for some of these things that are called cytopathic effects, and we can identify different viruses by this. So here's an example. This would be a situation in which the virus has caused host cells to fuse so that they can pass from host cell to host cell and get access to more nutrients to make more of itself. Okay. Another one would be where it's causing damage to the inside of the cell. The cell is not making the proteins that causes it to stick to the bottom of the flask that you're growing the cell in, and so they round up. That would be a cytopathic effect, or CPE. So this is how we can tell by looking either in cell culture or in a tissue slide, histological slide, where we can see damage to the tissues, and we can identify the viruses through the damage they're causing, the visible damage that they're causing. Now let's talk about pathogenic properties of fungi, protozoa, helminths, and algae. Since we've been talking an awful lot about the bacteria, we've talked about the viruses. Fungi cause damage by causing allergic reactions, and also by producing toxins. So they're kind of like the bacteria in that way. They produce exotoxins, which can damage the host. Protozoa cause pathogenicity, cause damage. They can replicate inside of the cells, damaging the cells that way. They can suck the nutrients out of the cells from the outside, kind of like protozoal vampires. And sometimes it's their waste products. Buildup of waste, which your body sees as toxins, causes disease by altering the function of the host. Helminths, or parasitic worm, can disrupt normal function That's elephantitis. Elephantitis gets into the lymphatic system. We're going to talk about that more in the next chapter. Blocks the flow of the lymph, causes the affected limb or organ to swell up to the point where it looks like it belongs on an elephant. That's why we call it elephantitis. Also, they produce waste products. They pee and poop into you. And also by stealing nutrients. That would be tapeworms. Now, algae, I love that they put cyanobacteria under the category of algae. Since they're not algae, they are bacteria, but we'll talk about cyanobacteria under the category of algae. They produce neurotoxins, okay? And there's, some of them produce blooms, red tide. You don't want to eat fish during a red tide because, and in that case it's not cyanobacteria, but it's still not algae, it's dinoflagellates. But they produce neurotoxins that build up in the fish. You eat the fish, those neurotoxins hit you and causes problems. Also, they're finding that some neurodegenerative diseases are caused by cyanobacteria. That is the same kind of thing, that you have a buildup of the neurotoxins as you go from the cyanobacteria to the fish. We eat the fish or we drink the water and it causes problems. Now let's move on to portals of exit. Now, there's three goals of a pathogen. They want to get into a new host, replicate themselves, and get out to a new host. Okay, so once they've caused disease, they need to get out. So they get out the same way that they got in, pretty much. And the portals of exit are linked to where they're causing disease. Okay, so for example, if you have a gastrointestinal pathogen, comes in through the mouth and it leaves through the anus, okay, through diarrhea. Sometimes it leaves back out through the mouth through vomiting. Okay, if you've got a respiratory disease, comes in through the respiratory system, goes out through the respiratory system, you get the idea. So those are portals of exit, the ways that the pathogen leave the old host and go to the new host. Alright, that's it for this chapter. Once again, this may or may not have been an easier chapter, but I suspect that it was an easier chapter because it's something that you've dealt with. All of us have been sick, unfortunately. So once again, feel free to go over this as many times as you need and come to class with any questions that you may have, or shoot me an email.